This is the second video of the ionotropic receptors lecture number 1. In this lecture, we will focus on explaining some basic concepts of metabotropic receptors. And you will see these in more detail in upcoming lectures from other professors. But I think it's important to mention what these metabotropic receptors are, so then you can appreciate better the differences with the ionotropic receptors that we will focus in the next videos. So, a metabotropic receptor is a type of membrane receptor of eukaryotic cells that act through a second messenger. They are also known as G-protein coupled receptors, or GPCRs. And they constitute the largest family of druggable targets encoded by the human genome, with more than 800 GPCRs in the human genome. And actually, more than 34% of medications approved by the U.S. Food and Drug Administration have GPCRs as their main therapeutic target, especially when it comes to treatments for neuropsychiatric disorders. So these complexes involved a complex of proteins that result in metabolic changes within the cell, producing a second messenger that acts from the inside to open an ion channel or elicit other intracellular events. The receptor protein involves or includes a transmembrane protein with 7 transmembrane domains. It also includes a G protein, and it will also include an effector protein that depends on the type of metabotropic receptors. For the receptor protein, it's important to mention that the N-terminal is located in the extracellular space, while the C-terminal is located in the intracellular space. This is a scheme of how metabotropic receptors work in general. There are different variations to this activation scheme that we will see in the next slides, but in general, metabotropic receptors work or they get activated when the neurotransmitter binds to the extracellular space to this N-terminus part of the metabotropic receptors. Binding of the neurotransmitter causes a conformational change in the receptor and activates a G protein that is located in the intracellular space and is associated with the C-terminus part of the receptor protein. This G protein is a GTPase GTPA hydrolase that requires the energy from the GTP to physically move away from the receptor and activates an effector protein. And in this case, it's an enzyme that catalyzes the generation of a new molecule or second messenger that acts in the intracellular space to mediate a signal that causes the ion channel to open, but also produces other cell responses that can modify protein phosphorylations and transcription. The G proteins associated to metabotropic receptors are heterometic protein complex composed by 3 subunits, alpha, beta, and gamma. In this scheme, you can see how these G proteins are activated in response to a neurotransmitter. So upon ligand binding, there is a conformational change that facilitates the exchange between GDP to GTP. In this process, the alpha subunit of the heteromeric G protein complex dissociates from the complex and it moves away from the receptor to activate the effector protein. Although this scheme doesn't represent what I'm going to say, the beta and gamma can also form a complex that can also dissociate from the receptor to elicit other functions. And then once the alpha subunit has executed its activity, the GTP is hydrolyzed again back to the GDP form, and the entire complex reassembles again. So G protein complexes can be very diverse because there are many different classes of G subunits. There are up to 23 types of different G alpha subunits in the human genome, about 7 types of G beta subunits, and around 12 types of gamma subunits. And the composition of the different subunits result in different G protein complexes with different functions. So the alpha subunits are classified into 4 major families. There are the GS subunits, GQ subunits, and G1232 subunits. They're all stimulatory, while the GI or GO subunits have inhibitory functions. And here you can see a structure of how a G alpha subunit looks like. So the composition of the different subunits and different types provides different ways of recognizing the effector molecule, but they all share a similar mechanism of activation. So metabotropic receptors can interact and regulate the function of ionic channels by either opening or closing ion channels. Or they can activate different effector proteins, initiating different intercellular pathways, such as those mediated by the adenylate cyclase or the phospholipase C. And these have different molecular mechanisms to control ion channel function and modulate transcription. So in this slide, we see a representation of how the adenylate cyclase system works. And in this case, this metabotropic receptor causes the activation of an effector protein called adenylate cyclase. This protein initiates the CAMP dependent pathway, which catalyzes the conversion of ATP into cyclic adenosine monophosphate or CAMP. Then in this process, the alpha subunit exchange GDP for GTP, comes to the effector protein, activates adenyl cyclase, and it results in the production of CAMP, which goes and activates a protein kinase called protein kinase A, which converts or phosphorylates different proteins and increases the concentration of ATP, which results in the opening of different ion channels. This process is stopped by a protein phosphatase that is adjacent to these channels that dephosphorylates the protein and causes the ion channel to close. Here there are is a summary of the most important things. So the adenylate cyclase system is activated by the G alpha S subunit. The effector protein is the adenylate cyclase, and the second messenger is the cyclic AMP. So what we've just seen is an example of how stimulatory receptors act through G alpha S subunits to activate the CAMP dependent pathway. However, as I mentioned earlier, different alpha subunits such as the GI have inhibitory properties. And if the metabotropic receptor is composed by these GI alpha subunits, they have a complete opposite effect and they end up inhibiting the CAMP dependent pathway. So when the GI alpha subunit is activated, it goes and inactivates the adenylate cyclase, which results in decreased CAMP levels and inhibition of the protein kinase A. And also the G beta and gamma complex that is associated to the GI subunits can also be liberated and activate downstream signaling effectors with inhibitory properties such as the G protein couple inwardly rectifying potassium channels. And an example of how these inhibitory receptors works, they're targets of some important toxins such as the pertussis toxin responsible for whooping cough. So this activation of another kind of G protein, alpha subunit, which is the GQ protein, activates or causes the effector enzyme phospholipase C to split a membrane phospholipid, PIP2 or phosphatidylinositol 4,5 bisphosphate, into a pair of second messengers. One is the DAG or diacylglycerol and inositol phosphate, IP3. DAG activates protein kinase C which in turn initiates protein phosphorylation. On the other side, the IP3 diffuses into the cytosol and opens calcium-gated channels, mainly in the nearby membrane of endoplasmic reticulum. And the calcium ions activate certain calcium-dependent enzymes downstream. It may also open calcium-gated potassium outward channels and chloride inward channels with excitatory effects. And here there is a summary slide with the different alpha subunits and the different effectors that they activate. And the different types of metabotropic receptors will be seen, as I said earlier, in more detail in upcoming lectures in this course. And here you can find, as usual, a short quiz that revises some of the most important concepts from this video.